So this is a continuation of our innate immunity discussion with a couple of questions highlighting some other aspects of this. So remember when we talked about how the body recognizes non-self, and this is a direct mechanism. So we talked about we have our pathogen recognition receptors, and those are going to be on your phagocytic cells. Your macrophages, your neutrophils are going to recognize PAMPs, pathogen-associated molecular patterns, okay? And so here we had our toll-like receptors, which we talked about, for example, TLR4 for lipopolysaccharide on the gram-negative bacteria. Those are your membrane-type receptors. You also, which we didn't talk about, you also have your NOD-like receptors, and these are cytosolic, within the cytosol of your macrophages, okay, of your phagocytes. You also have NOD-like receptors. Now, in terms of the pathogen-associated molecular patterns, we talked about how that would be, an example would be LPS, or it might be a specific type of DNA for a bad guy, this kind of thing. So that's the direct way that the body recognizes a pathogen, but there's also an indirect way, which we call opsonization. Opsonization comes from the Greek word to prepare to eat, okay? So the way that it was described to me by my immunology professor, Dr. Stanley, was that let's say that you have a pathogen, right? You have the bad guy, okay? And he has this sort of, slimy body, and the phagocyte tries to get a hold of that, and he can't quite grab a hold of it, okay? Almost like a greasy beach ball. So what happens with regard to opsonization, to prepare to eat, is you have these molecules that deposit, okay? Let's say deposit on this bad guy, all right? And what those do is those are essentially like handles that enable that phagocyte to grab the bad guy, okay? So if you remember when you get into microbiology that there are certain types of these guys that are slimy, okay, and they're very difficult to phagocytose without having this opsonization capability. So how does that work, okay? So there's essentially three handles, if you will, not happening at the same time, at different times. And one of them is C3b, okay, which is a complement protein, which we'll get to. Another one is C-reactive protein, okay, which is an inflammatory protein. And then you also have the other one, which is going to be your FC gamma, okay? So in other words, it's going to be an antibody. So let's say it's an IgG antibody, okay? So we could have an antibody that is essentially locked onto the slimy guy. You could have complement, which, like we talked about, or rather we will talk about further. Just remember at this stage that complement proteins make up, I'd read one place, something like 7% of the proteins in your blood, okay? And what these do in terms of innate immunity is this complement will just, by virtue of a process called tick over, which we'll get into, essentially just parks himself on the bad guy, and if a phagocyte comes by, or if it takes, if you will, this stays there, then this will become opsonized and will be phagocytized. And C-reactive protein, we know from inflammation, is that essentially it gets upregulated by a cytokine, IL-6, and we're just sort of slowly introducing these IL-6, and C-reactive protein is produced in the liver. So the important thing is that your phagocyte, okay, so let's say this is your macrophage, which we love, he has a receptor for each one of these, okay? So he has a complement receptor 1, okay? He has a C-reactive protein binding site, and he also has Fc gamma R. So he also has an Fc gamma R receptor so that he can bind essentially to that and phagocytose that. So this is an indirect mechanism by which the innate system is able to eliminate non-self. So the first question that I have was essentially a matching. So number one is, or the question rather is, match the correct phagocyte receptor with the following list of opsonins. Okay, so if you have IgG, which is the Fc, then number one is going to be Fc gamma R is A, and then B, C3b, complement is going to be CR1. So that answer there is B. And 3 is C-reactive protein binding site, of course, is C-reactive protein. Not particularly C, not particularly difficult there. Question 4, which of the following activates the NALP3 inflammasome? Okay, so the NALP3 inflammasome, let's just talk about for a second, is that essentially, this is obviously, as you can tell by the combining word, it's involved in the inflammatory response. Okay, so let's go back for a second. So we talked about how the phagocytes, the innate immune system, has the toll-like receptors, okay, and that's going to recognize PAMPs, okay. Well, there's another story to this, is that the pathogen recognition receptors can also recognize what they call DAMPs, danger-associated molecular patterns. Okay, so PAMPs are associated with pathogens. DAMPs, danger, would be something like within your own cell. For example, it could be some kind of necrosis, or it could be due to some kind of a silica in dust, for example. So there's other, essentially, that are not necessarily foreign in terms of being a live foreign enemy, but they can be just as toxic to the body because of their destructive nature. So the body will also recognize these and try to eliminate it, and that can be the source of inflammation. So let's talk about what the inflammasome does. Is that, let's say, for example, you have your toll-like receptor, right, on your phagocyte. Okay, he has a toll-like receptor. He recognizes a PAMP. And you go through, obviously it's an oversimplification, but essentially you go through a cellular cascade which activates NF-kappa beta, okay, which is essentially a transcription factor that will start, thousands of really cellular mechanisms throughout the body use this, including when you get to, pharmacology, it's particularly big. But this then will activate what we call pro-caspase. So you get this caspase, which you know from histology, which then, I know, long story, but important, is, well, then ultimately you will get IL-1 and IL-18, okay? So you get the pro. So what this does, the inflammasome, at the end of the day, it makes essentially this pro-caspase into the caspase that gives you IL-1, interleukin-1, and interleukin-18. Who cares? Well, the issue is, which we'll get to, is interleukin-1 turns on something like 3,000 different genes, okay, in terms of the immune system. And IL-18 is going to also turn on genes that are going to increase this hormone, if you will, this cytokine milieu, which is going to then drive the immune response and drive inflammation. Another thing that will activate the inflammasome is you can have these danger-associated molecular patterns, okay? And that's going to create the same type of cytokine, the same exact inflammatory process. So let's go quickly to the question, which is to say that which of the following activates the NALP3 inflammasome? So I'd say elevated extracellular glucose in type 2 diabetes. Yes, that does. B, silica dust. Yes. C, cholesterol. Yes. And D, monosodium urate crystals, which would be AKA gout. Yes. So the answer here is all of the above. And the important point being is that even sort of living bad guys can be just as inflammatory as these other, is some kind of a extraneous living pathogen. Number 5, which of the following binds iron and is one of the 4 lysosomal products that are released when the lysosome fuses with the phagosome? So quickly, the phagosome, I'm a phagocyte. I've got this little pod, and I capture the bad guy. I bring him in, and this is called a phagosome. Okay, and then that binds with the cellular, intracellular lysosome, and I form a phagolysosome, okay? And then that becomes this extremely cytotoxic entity for the bad guy. So that's what I'm describing here. So the answer is A, lactoferrin, which is the correct answer, which we talked about before. B, transferrin, which is simply, as you know, it's a transport protein for iron in the body. Nothing to do with that. Hepcidin, again, that has to do with iron metabolism, whether or not iron gets ingested or rather brought into the cell or not. And D is fenton. We know the fenton reaction, which is very reactive, but that's with regard to iron, but it has nothing to do with the lysosome's enzymes. Lactoferrin, A is the answer. Number 6 is, name the product of the armamentarium resulting from NADPH oxidase. Okay, so the armamentarium sounds very warfare-like. What is it? Okay, essentially that's what the phagocytes use. It's their arsenal, if you will. That's where the number or where the name comes from. So we talked about components of it. We kind of talked around the armamentarium, but we never gave it a name. So what did we have from the armamentarium? We have the lysosome. Okay, enzymes, which we talked about. We have NADPH oxidase, pH oxidase, right? We have myeloperoxidase, okay, and we have INOS. Okay, so these enzymes we talked about with regard to lactoferrin. We have the defensins, that group. NADPH oxidase, remember, ultimately it's going to give us hydrogen peroxide. Myeloperoxidase, remember, that adds chloride and we get hypochlorite. And then INOS is separate. It's separate from these guys, okay? And INOS is a inducible nitric oxide, not like eNOS. eNOS is something that, you know, is different. That's part of the vasculature that it, you know, gives you vessel, you know, stretches the vessels, lowers your blood pressure. This is INOS inducible, which is a very reactive oxygen or reactive species. Okay, so that's the armamentarium of the phagocytes. So the question was, which of the following are the byproduct of NADPH oxidase ultimately? Okay, and the answer is A, which we just went through, is hydrogen peroxide. All right. Now, number 7, what enzyme is responsible for the production of hypochlorite, which we just answered, which would be B, myeloperoxidase. And then number 8 is, what cytokines activate INOS? Okay, and INOS, again, inducible nitric oxide. And so we're going to talk more about cytokines, but if you look through the answers there, the answer is going to be F, which is TNF and interferon gamma. And it's probably too early to go into that, but it is important to mention that, for example, going back to pathology and immunodeficiencies, there are people who have an NADPH oxidase deficiency, chronic granulomatous disease, okay? There are people who have a myeloperoxidase deficiency. So what you can do for your patient is, if let's say they have a deficiency in this, you can try to increase INOS. And so you can increase INOS by actually giving them interferon gamma. And that's why these kind of questions are good to look at, because, again, it has a pharmacological solution to it. Number 9, what cytokines actually downregulate INOS? And so in terms of the downregulation of INOS, if you think about the immune system in general, we've got things that cause it to flare up and things that cause it to flare down, which is a good thing, because too much of a good thing is a bad thing. So in this case, we have the cytokines. The answer would be F, which is A, B, and C, IL-4, IL-10, and TGF-beta, which will make a lot more sense when we start talking about which types of cytokines tend to modulate the immune system versus which kind tend to upregulate it. So think of it this way. I like to think of it as like 10-4 good buddy. Okay, 10-4 good buddy means we've killed the pathogen, so 10, 4, and TGF is good buddy. TGF beta, I should say. And then the last question I want to talk about with regard to, well, we're on the topic of turning down the immune system, is we talked about the macrophages phagocytosing this bad guy. And then something we didn't talk about was the fact that once they get this bad guy and they start to process them, they start secreting these cytokines, which essentially start increasing the number of other macrophages so that you can keep the fight going. But again, there's a way to modulate these guys, and one of the mechanisms for that we explore in question number 10, which is CD200R on the cell being sent a negative signal or the cell sending the negative signal? Now this is a favorite question. So at the end of the day is, if I'm a macrophage, I have a CD200R, okay, I have this particular, receptor, and I think of R for retard. Okay, in other words, it's going to tell this guy to slow down, retard himself, go back to the donut shop or wherever you were. We don't need you for the immune system anymore. But who has the CD200? Okay, will bind to this. It has to be bound by CD200. Okay, that's going to bind to this and tell this cell to retard itself. So the answer to that question is going to be A. So CD200R is on the cell being sent the negative signal. Now what's fascinating about this is you might imagine that the types of cells, so cells that have the ability to regulate the immune system, are the cells that are going to have the CD200. We'll go through those down the road, but one thing I want to mention that's fascinating is that cancer cells, actually, some of them and some pathogens have CD200, which is a beautiful thing because it can tell your own immune system to retard itself, and that's one of its ways of being able to survive and be able to proliferate in the body undetected by the immune system. So these are just a few additional questions to section 2 in innate immunity. So question number 1 is, with regard to recognizing self from non-self, what do the cells of the innate immune system look for when ascertaining if an entity is self or non-self? So A is pathogen recognition receptors. Okay, no, because pathogen recognition receptors are what the actual innate cells themselves, so your macrophages, your neutrophils, for example, that's what they have. They have the pathogen recognition receptors. B, PAMPs, pathogen-associated molecular patterns. Yes, that's the correct answer. So that's what they're looking for in order to identify self from non-self. And C is haptens. Now what a hapten is, which we'll get into more with regard to antibodies in particular, is that a hapten is a little protein. It's a baby protein, but it's not a big enough protein to really elicit on its own an antigenic response, or immunogenic, response. So what happens is that a hapten will bind on to, for example, one of your cells, and then that will make a larger molecule that can actually be recognized by the immune system. I'll give you an example of a hapten. Again, not the right answer here, but an example of a hapten would be penicillin. Okay, so penicillin itself sometimes will lap onto one of your cells. Let's say, for example, a red blood cell. Well, that together will become antigenic. Your immune system will recognize it as a pathogen, and it will start a reaction, an immunologic reaction, and then of course you'll get problems associated with penicillin. So that would be an example. Oftentimes drugs can be haptens. Or if you wanted to make a vaccine, for example, you could fashion a molecule such as a hapten, lop it onto a larger molecule, and then you could make something that you could create a vaccine to. And we'll get into that with vaccines. On the second question, with respect to question number 1 is, what does the innate immune cell possess that enables it to interact with the foreign entity? And the correct answer there is A, the pathogen recognition receptors. So we already talked about that. Beat that dead horse. Number 3 is, which of the following is not capable of antigen presentation? So we haven't talked about antigen presentation to date. We've mostly talked about phagocytosis, recognizing self from non-self. So let's quickly talk about that. So you have your innate immune system. It goes around, it recognizes non-self, it phagocytizes it. It brings it into its phagosome, right? And then the phagosome fuses with the lysosome within the cell, okay? And then you start to kill the bug with your reactive oxygen species. Well, something else that happens, okay, and this is in the context, okay, let's say of an antigen presenting cell. Now, an antigen presenting cell will then process the guts, if you will, of this pathogen, and then it's going to display it on what we call an MHC class 2 molecule, and we'll get much more into this in a second. Again, immunology, as Dr. Stanley says, is not linear, okay? You're coming from all kinds of different directions, so just bear with me when we talk about something that we haven't fully introduced. So I'm, let's say, for example, a phagocyte, a macrophage, okay, specifically in this case. I bring in a pathogen into my phagosome, I fuse with the lysosome, I kill him, I process his guts, and I display it out into a major histocompatibility complex molecule number 2. I live, okay, to present that antigen to the T cells. Okay, that's essentially what I live for in this case. Now, before we get into that, let's talk quickly about who are the antigen presenting cells. Okay, so we talked about the macrophages, okay, the dendritic cells. The dendritic cells are a beautiful cell. They have these sharp processes. They won the Nobel Prize a few years ago, and they're very unique. We'll get into some of their unique aspects in a moment, but they are antigen-presenting cells and probably the most powerful of those that we have in the immune system. They're the darling of the immune system. And then the B cells are also antigen-presenting cells. And what this does, these guys are who links the innate immunity to the adaptive immunity. And they look at, what if we knocked out, what would it be better to have, the innate immunity or the adaptive immunity? And the truth is that you'll live longer with an innate immune system, but the other truth is that they need each other. They're mutually complementary to each other in order to be fully effective. So macrophages, dendritic cells, and B cells. And they're the antigen-presenting cells, and they're the ones that have this particular function, this MHC class 2 molecule, okay, which we will get to in just a moment. Let's go to the question number 4, however. What if phagocytes, let's go back, to question number 3. So we said, which of the following is not capable of antigen presentation? So the only one there is D, neutrophil. It's a beautiful phagocytosine. You get a heart attack, you blow up your heart tissue, neutrophils get in there, they clean up the mess. They're great. They're the first on the scene. They only exist for about 8 hours, but they are not a particularly bright group. They can't present to the T cells. They're not antigen presenting cells. Number 4, what do the phagocytes have that enable them to phagocytose a pathogen that has been opsonized? And remember, we talked about opsonization to prepare to eat. It's indirect recognition of the pathogen, okay? And we talked about C3b, we talked about C-reactive protein, and we talked about our, antibody FC gamma R. So we have FC gamma R is on a phagocyte, C-reactive protein binding site also on the phagocyte, and C3b, which the complement receptor 1, okay? And complement receptor 1, and just a quick note about that, it's also interesting, is that red blood cells can have as well complement receptor 1. I'll ask a question about that later, but just to quickly introduce that. All right. Now, number 5, what happens after a macrophage is activated? Activated means that essentially they phagocytose the pathogen, they get very, exercised by this, and they start to secrete cytokines, which we're going to get into, and that's the correct answer. Number A, cytokines are secreted. B, undergoes apoptosis, absolutely not. Undergoes mitosis. That's not going to be, mitosis is not going to happen due to ingestion of a pathogen. It is it starts to secrete cytokines. We'll get into the details of that in cytokines. Number 6 is, what's the purpose of the NALP3 inflammasome? So we went into that NALP ad nauseum a minute ago, but the point here is, just to review it, is that the answer is C, A and B above. It activates pro-IL-1 to IL-1, and it activates pro-IL-18 to IL-18. And before it even does that, another true answer on there was that it activates pro-caspase, okay, to caspase, which, if you were going to say which is the, sort of most important thing, because what this does is this is what takes pro-IL-1 to IL-1 and 18, same with IL-18 pro. Okay, so it's the caspase that enables you to split that molecule, or not split it, but to activate it into its active component. And IL-1 and IL-18 we'll get into, but again, these cytokines are instrumental, critical to not only innate, but also to adaptive. That concludes the questions on section 2. And now we'll go into section 3.